Sandbex. Hopefully, this will sustain for the next at least 10 years, as I anticipate. Sandbex, carved out from Landbex, which we have been doing over the last, I would say, 4 years, called as Lankan Bioinformatics Conference. And this evolved, way back in 2017 and 18, when I went to Sri Lanka in organizing a couple of workshops upon their invitation. Lankan Ministry and University of Colombo, they felt that bioinformatics is its infancy, and they felt that many students from Sri Lanka coming to India, and they needed the help of Bioclues society to ensure that bioinformatics can be taken up to the next ladder. And with a very big hope, we tried to raise an MOU. At that point of time, I was at Birla Institute of Scientific Research, and I always keeping any conflict of interest. My bosses were very nice. They always obliged to my request in organizing conferences with Bioclues, and I took that opportunity with only one particular selfish agenda, that we should have we should educate students with bioinformatics, or we should take bioinformatics to the next ladder. And at that time, I was already involved with AP Bionet. By then, I already have been associated with AP Bionet since 2004. And then, of course, we already connected them, to those particular people. Some of the members became AP Bionet members as well. And some of them, went on to become the Goblet members. Some of them even went on to become the ISCB members. These are all the so-called, big societies. But one thing, really pains me every time, when we South Asians, practice bioinformatics. I mean, it is believed that 1 in 3 students go to either UK, Europe, or US, or for that matter, Australia, and New Zealand for their higher educational studies, including bioinformatics. And if they were to go, the main agenda is, of course, to learn, to earn, to gain some kind of an experience, settle there, and of course, give back, to the alma mater, to whatever, best, that they could do, so that, a new budding entrepreneur, can come to the fore, so that, they can take, bioinformatics, to the next generation. But, from our surveys, that has really not happened, unfortunately. We have been, mentoring graduate students. We have been, helping them place across, different spheres, not only educating in the form of, virtual projects or small, tidbits about their bioinformatics installations or their, career growth, or even for that matter, for any kind of, admission processes or recommendations or whatever. But somehow, that has really been in a very, 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 abysmal situation. So keeping in view of this, I thought that, we should have Sandbex, which means we'll take, 3 or 4 more countries, largely, South Asian countries or Southeast Asian countries as well. Southeast Asian countries, we thought, we'll make it in the next level. But at the end of the day, the main target is to ensure that, it's the global South. Global South is nothing but all the countries in the Southern Hemisphere. If you see all the countries in Southern Hemisphere, excepting probably to a smaller extent New Zealand and some regions of Australia, all other countries are all developing countries. They're not really developed countries, per se, excepting maybe Singapore. Maybe they might be there, but those countries have got very good bioinformatics infrastructure, bioinformatics development. But other countries remain absolutely very, very poor. So, keeping in view of this, we thought in a very modest way, why not, we come up with Sandbex, South Asian Network on Bioinformatics. This has been in our mind over the last 1 year. We have been fervently requesting several people. We shot at least 200 to 300 emails, met several people in conferences. But somehow, that interest has not been, with due respect to them, that interest has not been that great as anticipated. But I thought, with my good friends like Ashutosh Das, who is my postdoc colleague in Denmark, who has come back to Bangladesh as a professor to serve his country, Likewise, I've got some friends in Nepal, some friends in Pakistan, Ilyas, some friends in Sri Lanka as well, of course, from the workshops that we organize. In fact, several of them visited my lab while I was in Jaipur when we were, organizing. But none of them, unfortunately responded to the emails. This has really been, hurting me. And to be very honest with you, I've had some kind of, sleepless nights as well, because we're not doing any kind of business here. We're not doing any kind of this thing, accepting with one thing, one notion in our mind, to ensure that we take bioinformatics to the next level, be it in the ambit of Bioclues or be it in the ambit of any other organizations. But at the end of the day, we should ensure that Bioclues, bioinformatics, should really win. That was the only ray of hope that we had. Okay, so with this conference, we thought we will announce this sandbox. I hope all 41 registrants here. I thought even if there are only one or two also, we will go ahead, because I'm a bit surprised. I'm just trying to understand the system. There are over 9,500 members in Bioclues, and as you could see, we are only about 23 today. Okay, and we had about 89 registrations, and 83 minus 29, none of them could really attend this today. I mean, I'm a bit probably surprised, bit miffed. I'm also concerned about the discipline. I'm also concerned about the etiquette, demeanor, and how we could really ensure that we take it to the next level. So we'll have a rigorous brainstorming panel discussion sometime the end of the conference. But now it is time we started our conference. So we have four excellent speakers in the form of Ashutosh, who will be introducing his own research besides bioinformatics in Bangladesh. That will be followed by Murali, my colleague, and friend. He has been doing wonderful work on genome proteomics as a proteomicist, as a mass spectrometry specialist. His group has been doing tremendous works on snake genome project across South India, and as well as some parts of Sri Lanka and all those kind of things. I hope this will really bring up that kind of scientific camaraderie. And then we have Muhammad Ilyas. I was a big fan of his article in PLOS Computational Biology, where he has written on bioinformatics development in Pakistan, or computational biology development in Pakistan. He went to UK and then he returned back to India. Again, he wanted to serve his country and ensure that he wanted to educate masses. So he's the one. I've been trying to connect to bioinformaticists in Burma, Bhutan, Nepal, and Sri Lanka, but some of them they couldn't turn up to my requests. So but Sri Lankan participants who are here, they will be a part of a rigorous brainstorming panel discussion. So without much delay, with two folded hands, I would like to welcome you all, and thank you all for coming all the way across different time zones. And we also have Asif M. Khan, who is the president of AP Bionet and the president of Goblet as well. So we have been a part of Goblet as well. So we thought we will have their particular talks and ensure that we take it to the next level. So without this thing, so let's welcome Ashutosh Das. So Ashutosh Das will be introduced by, I believe, Jayaraman sir. Jayaraman sir, please. Yeah, over to you. Thank you.
Raj, I was introduced Murli.
Oh, so sorry, sir. Sorry, maybe Sundarda. I'm sorry, I was confused. Sundarda, please. Yeah. Sundarda is also traveling. He was in the flight, some time ago. Yeah.
No problem. I was in India. As long as I was in India, I was part of bioinformatic network. Many of you may not know. When it was started in 85, late 88, like that. I was the first person joined the bioinformatic network. But later, after 10, 15 years working in Calcutta, then I went to do my PhD and all. So I was very keen to know how I know how the India has developed the bioinformatics, how things have happened, how it is dimensioned in various sub of bioinformatics aspects. But I was very keen when I see the title of the talk from from Bangladesh. I was really impressed. We want to really know what things are happening in Bangladesh, our neighbor country, which I was very closely attached when I was in Calcutta. So we welcome Professor Ashutosh Da. In प्यार से we say Ashutosh Da. Is Ashutosh Das, Professor of Genetics at Chachogram Veterinary and Animal Science University. In short, we say CVASU. He had his PhD in Denmark, as Prashant was mentioning, in genetics in 2015. Later, he had postdoc in Australian National University, and he is doing a lot of things in a variety of bioinformatics like genomics and metagenomics, evolutionary genomics, microbes, and things like that. So we are very keen with all other participants to know what our things happening in Bangladesh. So we welcome all to hear from Ashutosh, and later we can have at the end question and answer, and we can collaborate with him and things like that. Welcome, Ashutosh.
Thank you, Sundarajan. Can everybody hear me? Because we have,
Yes, please.
we have some internet going on this week, so I hope you can hear me fine. So before going to my presentation, I want to thank Pras to having me here and welcome everybody to PICS conference. So before going to my presentation, just to share. So, share. Sorry. I think you can't see my presentation, right?
No, Ashutosh, you have to click your presentation slide now.
Yeah.
And then make it full. Yeah, now we can see. You can make it full screen. Yeah.
Can you see that?
Yes. Yeah. Maybe it's in a slide.
Yeah.
It's fine. Please go ahead. Yeah. Thank you.
So it's okay right now, right?
Yes. Yeah.
Ashutosh, you can click the full screen. The bottom, the full screen, then it will come without the notes.
Okay. So you see my screen, right? No.
Yeah.
Okay.
Yeah, now it's good. Thank you.
Okay. So talking about Bangladesh, and I will go through a very brief scenario and perspective here in Bangladesh. So as I introduced Sundarajan, I'm working in Chattogram Veterinary and Animal Sciences University as a professor. So if we talk about the kick of the play here in Bangladesh about the bioinformatics, it's mostly overseas collaboration based. So first time it came into light when Bangladesh declared about the decoding of the George genome sequencing, and followed by decoding a crop killer fungus in 2012. So there are some foreign collaboration, like I was doing PhD and some students and early professor was doing their PhD and postdoc overseas. So we do some characterization of animal genome and analysis of big genomic data, something like that. Followed by when we come back, like researcher like me, and we start doing some nation-based training programs to give some dissemination about the bioinformatics and computational biology work. And we've gone through several training program workshops throughout the country, mostly in the Dhaka University, University of Technology and Science in Chittagong, and Asian University for Women. It's mostly occurred during 2016 and 2017. So after that, there are some, institute came forward to establish some center for bioinformatics and learning advancement and systematics training in Dhaka University, something like our Bangladesh University of Engineering and Technology. There formulated a research group based on the bioinformatics and computational biology. And North-South, a private university, North-South University, they established a genome research institute over there. And Rajshahi University is a big university in Bangladesh. They also formulated a research group called Bioinformatics Research Group over there, and Genome Center in Joshua Science and Technology. So these few are some research group has been established over the period, but we got in the outbreak of COVID-19 here in Bangladesh about the bioinformatics, particularly doing the so Bangladesh established a research lab at that time after COVID, and also IRV, that very good genomics, also a computational lab over there, and Child Health Research Foundation came forward with the genetics and bioinformatics, and also our National Institute of Biotechnology, they formulate a genetic data with bioinformatics. And also, like the last but not the least, the university, we established a lab. I am in charge of this lab. And so talking research group and also about the lab, but we cast about the academic anchor, the Bangladesh Agriculture University there. Just start a BSc degree in bioinformatics and engineering. University I started a BSc degree in molecular medicine and bioinformatics. In my university, we established a called Genomics and Bioinformatics, and here Asian University for Women, they introduced a course like introduction to bioinformatics with lab. So there are some more, but I'm missing some of them. But anyway, we are at the at the starting point of the academic program here in Bangladesh. So if we talk about the area of focus, what have been done in last decade, genomics was the key focus by decoding jute genome, hill surface genome, black Bengal goat genome, red Chittagong cat, genome, jackfruit, white Bengal tiger, SARS-CoV, and so many pathogens. So pathogens, there are more genome decoded here. If we talk about the transcriptomes, is but not that much done here in Bangladesh. Talking about jute, hill surface, dengue virus transcriptome, and proteome, I think it's a very few work have been done here. Proteomics, particularly proteome, foot and mug disease virus, proteome, something like that. One is the microbiome. It is a sparing area. Bangladesh already. Some people have done the human gut microbiome, Hilsa fish microbiome, roe fish, and bovine mastitis, something like that. So if we talk about the component of the bioinformatics area have been done in Bangladesh, we talk about the majority have been done on the analysis of the data computation. So something, including me, I'm doing something in that line. But for development of the algorithm and statistics, this is mostly centered on Bangladesh University of Engineering and Technology, CSC department, the CC department of Islamic University in Kustia here in Bangladesh, and one more university, Rajshahi University, that is a statistics group. They are doing something on algorithm and statistics on bioinformatics. The gray area for Bangladesh is the creation of the database. It's remain as a gray zone here because I haven't found anyone working focusing on that part here in Bangladesh. If I talk about strength of Bangladeshi bioinformatics situation right now, we do have several research groups, resources, several genomics labs, and some academic programs. In my opinion, I have found some shortcomings here. Is mostly the computational infrastructure. I'm studying at one. Having a good training research from Europe and Australia, I'm having computational facilities here in Bangladesh. The talk about the collaboration is still limited with the very few people, not in the nationwide collaboration. Also, another one is the effective bioinformation is missing here. We do have two. They're like in the name, not what informatics and focused on the bioinformatics and to get together in a every year in a workshop or thing like that. So if I talk about the prospect, the many unexplored area of bioinformatics having here functional genomics, metagenomics, oncogenomics, drug development, disease biology, health, evolutionary biology, metabolomics, big data mining, system biology, many more. A very good place to start with here in Bangladesh. So, very brief on Bangladeshi scenario. So I will talk about my work adding the pipeline, getting Novo assembly. Here are some few of my regarding genomics and bioinformatics here. I will talk about one of my particular work I have done with the collaboration with National University. So we plan for something novel, the nuclear genome assembly of Eucalyptus passiflora as a I just wanted to know, can because going on. Hello.
Yeah, please go ahead. Please go ahead, Ashutosh. Yeah.
Sure. Yeah.
No, it's all well, Ashutosh. Yeah, please. We are eagerly. Yeah, please.
So, our give line for a comparing assembly. The technology for the short read to having a genome assembly, but these days we have third generation technologies like MinION, SpecBIOS, and some more, Hi-C. So when we do it assembly with the short read assembly, and we get very high accuracy, but that is very fragmented genome, you would not get any information, maybe less information on the structural variant or structural heterozygosity of that genome. So for that, people are using the long read, having the genome assembly. But if you having the third generation technology, we are getting more complete genome. But the problem with the accuracy, whether if you think about you have the 5 or 10 genome assemblies from the same data using the different algorithms are having change even in single parameter, you will get a different genome. So, there are still many challenges remaining to get a better assembly. So, in our work, we try to very, I'm not saying perfect, it's a good one, doing something, choosing a set of possible. So you need something like a measurement. So commonly we do commonly use the N50 and BOSCO for the genome assessment. This is very two common metrics. As you know, the N50 is measuring the contiguity, not the accuracy. So it is sometimes artificially inflated by the incorrectly linking contigs. And if we go for the BOSCO, it measures accuracy, completeness of the genome assembly, but it's only assess the small portion, the less, the coding sequence is less than 1%. They assess less than 1% of the assembled genome. So you need a range of metrics to assess the compare genome assemblies. So in our case, for the snow gum, we call it Eucalyptus passiflora. It's a very common Australian snow gum tree. So we think about that, and we start our work with. We propose a very it's a novel, but it's a measure for assessing the genome assembly. The assembly, we call it assembly ploidy. So it is the proportion of the genome that is represented by the haploid. If you want to deploy polyploid genome, how much that is represented by the haploid contigs. So we call it assembly. So if we go for the, in general, we talk about the like heterozygous problem. People go mostly about to elevate the problem. People go mostly about the inbreed individual to get a better assembly because there is low heterozygous region, and a very good algorithm can easily collapse those reads in the haploid sequence. But what about outcross or very heterogeneous individuals when you go for that particular for the plant? So we because you will get two types of haplotypes, but they are particular. If you get heterozygous, you will get the primary contigs that is referring, because for the same region you will get the two contigs. One is primary contigs, one is haplotics, referring as a haplotics. The longest, the longer two of the two contigs is called primary, and shorter haplotics. So for our, maybe it's imperfect, but we assembly ploidy. If you assembly size and estimated genome size, you might get an idea for representing the haploid diploid. So if you put size in the numerator and estimated haploid in the denominator, I said it's sensitive in the estimate haploid genome size, and also sensitive to the area of genome assembly, particularly the highly incomplete assemblies that might affect the numerators. So but nevertheless, in combination with the other measures, we show, you can see in the next slide, this assembly ploidy provides a useful information for comparing the genome assemblies. So as you know, people are using several other matrices for genome assembly assessment. If we call the Seagull computing genome assembly likelihood, a higher likelihood that suggests the represented by the throat, also the LTR, long terminal repeats, a higher LAI score suggests a more contiguous and complete assembly. So we also use this in 8 measures, including 50 Bosco assembly, Seagull, score, and we also use the base level error rate. Traditionally people use the remapping the same set of reads to the assembled genome. Where they did the genome assembly, they also use the same set for the here we did not did that. We just to avoid the biases. So we integration that are not biased, but from the same individuals we sequenced. So we use 10% of that, and so we found that slightly decreased the biases using the same sets of the assembly sets, remapping things. So we also use the number of structural variant when remapping the wrong read to our long read assemblies. Like, as you know, if you have the more structural error rate, that indicates the assembly has a large proportion of the incorrectly linked contigs. So we also have done that. And the final measure we use, that is the genome sequence similarity of each assembly when we compare with all other assemblies. That means when you have a set of assemblies, we remapped with each other. That is not giving the information relative to the underlying truth, but it is significant, to get the information about the best plausible assembly. So if I talk about the data, we have sequenced eucalyptus trees that is 5,000 years old from the Thredbo, New South Wales, Australia. So we generated 2 data sets. One is long read data sets, one is short read data sets. And after filtering, we got 193x for long read data and 246x coverage for the short read data. So measuring, just assuming the genome size of 500 MB from the flow cytometry results. So when we did it in silico genome estimation, we use the genome scope that is using the 32 MAU. It gave a very low, like underestimation is like, you can see this 408 MB. So it's almost 100 MB difference. So what is happened here, the genome scope tools cannot perfectly account the repeat content, because it is a heterogeneous species, and also we found that the repeat content of the Eucalyptus grandis, the previous published the grandis assembly from the US, they found it is 50% repeat content. So we use another one like SGF Prexy. So it's better account for the repeat content, and we got for 529, so it's almost close to the 500. So we thought, we are getting the right, we are in the right track. So as I said, we did two sets. One set is for the assembly data sets, and we used average 10% of the long reads and 7% of the short read for the validation sets, and like 222x short read for the assembly, and also long reads for the assembly. So we use 5 different assembler for long read only assembly: Canoe, Smart De Novo, Fly, Marble, and we also use a Masarka as a hybrid assembler. And we divided our long read into 2 different category. One is the minimum length is 1 kb, and we also cut up another sets called is 35 kb. Minimum read length is 35 kb. So for each case we use these 5. So it should be like 7 long reads, only 2 hybrid assemblies, but it should be like 10, but here we got 9, because this Marvel is very computational. You need a more computational space. So for Marvel, we only did the 35 kb one, 1 kb one. I could not even done it with the let's say for the computational facilities from the Australian National University. So anyway, we will go for we've gone through like 9 of the assemblies. So when we did the assemblies statistics with the first, we found that for the 9 assembly, the hybrid assembler, it seems like that is giving the better N15, and for the long read only assembler is giving the Smart De Novo is giving the longer N15. So this is the beginning of our assessment. So while doing this, we think about whether, as it is planned, whether we are getting the right DNA or right reads for our assembly. So we gone through the contamination detection, and initially we used the all reads mapped with the blast, and after that we use blob tools to assess the contamination of it. You can see the Streptophyta. The plant is like most of the almost 100% heat of the, some of them are close to 100%. So we could not find any other heat. So we are in the right track. So we got the genome, and we got the N50. So we start the polishing the genome. We used Recon for long read assembly using NGMLR, and after that we use the Pylon for using Butai2 for the short read assembly data sets. So for Mizarka, we could not use the Raccoon one, because using the Europhone long reads polish with the hybrid assembly, it tends to induce more errors rather than remove them. So so you got this idea, like we used Raccoon long read, and for Pylon for the short read. So going for the assembly ploidy and haplotic removal, we use parse haplotic here to get the idea what are the primary contigs, and because this primary parse haplotics is divided the contigs into which one is the primary one and which one the overlapping in the same region, so giving the haplotics. So after each stage of the polishing, we are getting the better, let's say here assembly ploidy, because we want to represent the diploid assembly in a haploid manner. So you can see the assembly is close to the diploid stage. So we are getting more iteration for the Raccoon and Pylon polishing, and getting lower and lower, and here you can see it's close to 1 in some cases. So very polishing stage, and after polishing stage, so it's giving more removal of the haplotics and giving the more closer haploid genome assembly. So here are some image figure about the Bosco and total structure variant. So we did some, like, after polishing, we did Bosco analysis for complete complete genes. Also did the duplicate genes. So you can see here you can see the Misarka one is and also like it's getting better here about the hybrid assembly. So for the complete Buscogen, sometimes it's giving the decrease and sometimes it's giving the increase. So it is a bit confusing for the Bosco for in some case. So the problem here is making about the repeat region. So that's why we go for the more one is called the total structural variant. So you can see here for the canoe, this star de novo fly and everything you can see here in Misarka giving them less structural error here in so this is the final one. We talk about that was the genome sequence similarity. We mapped all the assembly with each others, and we try to get the idea where is the genome sequence similarity here. And you can see the Mazurka there, like almost 100% sequence similarity, and getting a better result with the most of the genome assemblies. So we use here the NUCMAR from the Marmar tools. So talking about the all measurements in the same scenario, in the same picture, like N15 Bosco score, Lie score, assembly ploidy, base level error rate, Seagull score, structural variation, and genome sequence similarities. So you can see.
Ashutosh, how long? Because we have already passed 8 more minutes. Yeah.
Okay, it's done. It's done. It's that last slide. It's okay.
Yeah, Ashutosh. Yeah, please. Hello? Please. Yeah, please, Ashutosh.
Yeah, please continue.
So if you think about the context size here after the polishing and everything, we got much more better N50 here in Misarka, and also for the complete gene in Bosco analysis here, and talking about the Lie score here is Misarka is very good one, and also the error rate here and the error rate for the long read mapping. So altogether, we found like Mizoka 35 kV set was the better one. So not only using the only algorithm getting an assembly, we always prefer to get a more measurement to get a better assembly. So here is my link for my papers, and so I do have some ongoing research as functional genomics on captive tigers, Archidong, Sivas, mogulfish, a few more. Actually, I'm looking for a collaboration, particularly in the infrastructure facilities. If someone from this conference want to collaborate, I'm happy to collaborate. And thank you for listening me here. Thanks.
Thanks, Ashutosh, for a wonderful intro and the very good work you are doing. And we love to have collaboration because that's the purpose of this Sunbake. And we do have a lot of kind of collaboration like children level, school level, all the levels actually we are doing it. So we are going to do that. Anyone from the audience has any question to Ashutosh? One or two quick questions. Huh? Yes, Prash.
Yeah, thank you, Ashutosh. Thank you so much, for the wonderful talk. I mean, I understand, when you were referring to haplotypes, why would you talk about, heterozygous calls? I mean, for the inbred lines, you would always expect to have homozygosity, right? So and
Yeah,
yet, why would you talk about, I mean, why would we talk about the heterozygous calls at this point of time? Is it
yeah.
only a statistical inference, or yeah?
No, no, it's not only a statistical inference. It's particularly, for plant or for animal, when you have the option for the getting inbred, it's okay. But talking about the plant like in the wild, as we done for the snow gum, so that is very outcrossed. So that is very heterogeneous. So I give the reference here for the haplotics because of the sometimes you need to assemble the genome that is you can't find the inbred population. So in that case, you need to consider about the haplotics. If we get the let's say you have a father, mother, and the child. So you sequence them and you get the better haplotype over there. So it's very easy for the control breeding. For something like you are getting something from the marine, you don't know about this. It's very heterozygous maybe. So in that case, I refer the haplotics removal for that things.
Right, wonderful. Thank you so much, Ashutosh. Thank you so much amid your busy schedules and chaos in your university campus. You have made it. Thank you so much. Wonderful talk. Friends, any question for Ashutosh? I still remember, when I joined as a postdoc, he was just wrapping up his PhD. I had a very short interaction with him, but ever since, I've been, trying to stay in touch. He has been doing yeoman service, in Bangladesh, and he wanted to be there in Bangladesh and take up, bioinformatics, to the next level, and all those, third generation sequencing that he's talking, and all those, wonderful technologies he has really, introduced. And I think, as an animal geneticist, he has been doing a human service, to Bangladesh and largely, South Asia. So if there are no questions, I think, Sundarda, we can go to the next speaker where Jayaraman sir can. Yeah, I think there is a question from someone. Sundarda, you can attend. Yeah, Santosh has a question. Yeah, please.
Yeah.
Sir, about this data cleanup you told about, can you suggest any freely available packages so that we can go for data cleanup, or we have to depend on only these proprietary softwares? We
For the
cannot
data,
afford these packages.
for the data cleanup. You are talking about I use the
You use
something like that.
Yeah.
Yeah. There are some free for what you are normally using right now, because there are some data cleanup you can use.
We have sequence reads from Ion Torrent platform.
Okay, so for the short read, you have mostly the old version, because these days you can use the thermometrics, and also like some are like that's thermometric is the good one, as I said, because for the short read. And I'm not exactly, I can't remember the exactly there is something very specific for the iron turn, but
If I can add a comment here, Ashutosh. Santosh, what I understand is, iron torrent has got, different capacitance and dimensions. And because, Dharma has acquired it, so what has really happened is, they have their own cloud-based server, and they have their own inherent tools, through which, they run, actually. So even the tools like Masurka, Dibika, these tools, they do not probably, run over there. So you have to use their in-house stationed tools through cloud. Yeah. Thank you for the data cleanup. Yeah.
I think it's called Partech. Their pipeline in Iron Torrent.
Yeah, Partech. Yeah. Pushpendra. Yeah. Thank you. Yeah.
Thank you, Ashutosh, for a wonderful intro. We now welcome Professor Jayaraman to moderate the next section. Thank you, Jayaraman, sir.
Good morning, all. Please mute. Raj, please mute. Yeah. I am very pleased to introduce Dr. Muralidharan. He is currently serving as an assistant professor at the School of Biotechnology, Amrita Vishwavidyapeetham, Amritapuri. He has a PhD in biotechnology from Amrita Vishwavidyapeetham, and his research mainly focuses on the characterizing of the venom proteome and assessing the antivenom immunological cross-reactivity of medically important snake species from the Western Ghats of India. Earlier he worked as a research fellow at the Amrita Center for Nanoscience and Molecular Medicine. He was also associated with the Amrita Center for Research Development as a research consultant, and he was engaged in various consulting services. In this role, he was involved in developing multiple qualitative and quantitative molecular characterization workflows for proteins, peptides, and therapeutic metabolites using HPLC, mass spectrometry, and related tools. He currently plays a key role in successful functioning of the core mass spectrometry facility at the school. His research includes snake venom proteomics and natural product-based drug discovery. I am pleased to. Today is going to talk on from transcriptomic to proteomics, the importance of integrating omic approaches to snake venom characterization. I now request Dr. Murali to come and give his speech.
Thank you so much, Jayaraman sir, for the introduction. And first of all, I would like to thank Dr. Prash for inviting me. So I think this is the first time I'm giving a talk to a bioinformatics community, and I'm happy because majority of the work that we do focuses on analyzing the mass spec data using different bioinformatic tools and softwares. So I would say majorly means 50% of the work wet lab. I mean, I all of you know that. And